Okay, we're going to start off now talking about one of the more important polymers in the universe. We're going to talk about DNA, and more specifically, we're going to talk about the parts that DNA is made up of. So, as I said, DNA is a polymer, and that means it's made up of many sort of repeating pieces. So what I'm going to do today is I'm going to draw over here on this side of things, I'm going to draw a sort of diagram of what DNA looks like. It's different little pieces. And then over here on the other side, I'm going to try to draw out the actual chemical formulas. And the most important thing to remember is that this is not that important, the chemicals over here, but it can help you just sort of remember what's going on and why things are named the way they are. And this is more meant to sort of inform your learning, but this over here is what you're really going to be working with. And one thing that I really want to emphasize today is that thinking of DNA as a polymer and understanding this diagrammatic structure of DNA is going to be really helpful as you start doing certain techniques in the lab. In particular, PCR, which is one of the things that we're kind of working towards having a really strong understanding of. And I say really strong understanding of because this is a pretty simple process that you don't have to actually completely understand to be able to do. But once you've mastered exactly how it works, you gain an ability to tinker with it and do some new exciting things and understand why things go wrong when they go wrong. Anyway, that's just a motivation for why I'm telling you this in the level of detail that I'm telling it to you today. Just try and remember that this is all going to come in handy. Alright, so DNA you probably normally think of as this sort of double helix. You know, you have two strands of something or another going around, and somehow they're sort of linked to each other by these base pairs. And what are base pairs? Well, who knows? I don't know. So today we're going to talk about, unzipping this, taking one of these strands and just talking about what it's made up of. So we're kind of zooming in on this one strand and we're going to talk about what it is. So DNA, very simply, is a repeating polymer of either deoxyribose, backbone, with attached bases, or it's a repeating ribose backbone with repeating bases and phosphorus linkages. And that all sounds a little weird, but let me just draw what that means. What that means is that you have a specific kind of chemical, it's a kind of sugar, called a ribose, and just replace this with deoxyribose if you're thinking about DNA. You have a ribose or deoxyribose here. That links by a single chemical bond to a phosphate group, right? And this is the phosphate group. And that, in turn, links to another ribose group. I'm not very good at drawing circles, but another ribose group, which goes to another phosphate group, which goes to another ribose group, which goes to another phosphate group, on and on and on in both directions, a heck of a long way, and until it's about 2 meters long worth of this stuff. And at every ribose branching off of it, there is a base, right? So there's this strange structure, we're not going to say exactly what it may or may not be, that is called a base, and or it is a base, and these things are either a uridine, thymidine, cytosine, guanosine, or adenine. And the reason I drew these two on top of each other is that they're kind of the same, but this one is for RNA and this one is for DNA. For some reason in the RNA world, when you're dealing with RNA, it uses a U, and in the DNA world it uses a T. And I have no idea why. Maybe somebody can tell me why that is. It seems like you would just want to use one. It seems like a lot of trouble to make both of these, but whatever. Excel has a reason for doing most things, and it probably has a reason for doing this. I just don't happen to know what it is. So at any rate, this is your basic story. This is what one backbone looks like. these are the little bases coming off of it. These are the side chains. And that's your story. That's DNA. But let's look at what the chemicals actually are. And the real reason I'm telling you this stuff is just so that a little bit of the nomenclature later makes more sense, and a little bit of the details of how PCR work will make sense. And much, much later when we talk about sequencing, and in particular when we talk about a kind of DNA sequencing called 454, there's going to be a very particular little detail of this side of the equation that'll be interesting and important. All right, so let's switch colors.